Here we are.
Okay, so I will start. So good morning, everyone. My name is Alessia Carrara, and today I'm going to present my master thesis, which is entitled as Machine Learning Classifier for Acinetobacter baumanii plasmids. First of all, I want to thank you, you, Adrian and Patrick, for sure, and also to let me present my work here, and especially the BioIT group with Sergio Redondoloso and Anita Schurj, which are my supervisor, and also my additional supervisor from the University of Milan. Let's introduce the bacteria species on which the work is based. It's Acinetobacter baumanii. It's a gram-negative bacteria, mainly found in a hospital environment, where it affects people with compromised immune systems. And several are the infections for which Acinetobacter is responsible. According to a Rubik et al. Study of 2018, as we can see in this graph, I hope you can see my mouse moving. So according to among 125 observations, which are distributed according to the sequence to the type of infections, Acinetobacter baumanii resulted to be the most abundant species. So it is clinically relevant, as we can observe here, in abscesses, pneumonia, septicemia, also urinary tract infection, and also wound infection. It has been classified also as an escape pathogen due to its resistances to antibiotic treatments. And as we can see here in the map, we have several resistant Acinetobacter baumanii isolates, which have been reported actually worldwide. As background information, moreover, I would like to introduce the importance of the genetic content of an isolate and the problem of retrieving the genetic sequences. To do this, I will be helped by these Mondrian pictures. And the collection of the pictures could actually represent the content of an Acinetobacter baumanii isolate, as well as any bacteria isolate. A bacteria isolate has a chromosome, as we can see here, and then it may have or may not have one or many more plasmid sequences. In our case, it has 2 plasmid sequences. The most popular sequencing technique to retrieve the sequences is whole-genome short-read sequencing, which generates short reads, which could be identified as small pieces of puzzles of these pictures. However, for this method, it is impossible to completely reconstruct the genetic sequences. This is due to, for example, the repetitive sequences within a single genetic element. As we can see here, the piece could be located in different locations. Moreover, it can also be shared, it could be a gene or a contig, which is shared among different genetic sequences, like a chromosome or a plasmid, as we can see here. So the assembly problem could be solved using actually a bigger pieces, so that using long-read sequences. However, this technique is not so popular, and also short-read sequences remain the technique which is most used. So we consequently we should try to take advantage of short reads. If we look, as we can see at their backs, we can observe that the chromosome painting is colored in light blue, while the other 2 are colored in orange. What we tried to do was to take advantage of short reads of isolate, of which we have a complete access to their content, so that to the composition and also to the nucleotide sequence, to generate a model able to classify if a group of pieces, so that a contigs in the end, is part of the chromosome picture or of the collection of the plasmid pictures. So, but however, without identifying which one of the plasmid pictures. The approach we use is a machine learning based approach. And in our case, we work with a binary classification, so that plasmid and chromosome categories. However, generally, a machine learning approach consists in generating a classifier, as we can see here, such as a small program in the end, based on a set of initial data, from which we extract some features, and then we use an algorithm which is trained on them. The output classifier is then subsequently used to predict if a new set of data, here, described by the same set of features, belongs to a class or to another. The extraction at this point, extraction of the features is a really important step in machine learning. And it consists actually in finding some properties that specifically identifies one class with respect to the other. In our case, in this case of the collection of pictures, we can assume that we can mix all the pieces together, and we try to identify if a random selected piece is part of the first picture or is part of the second or third picture. And we can expect that, for example, pieces like these, which are colored with just one color, are part of the picture one. While when we have a composition of color, for example, in a single piece, it will probably be part of the other two paintings. Similarly, we can also consider another kind of features, which is the amount of white color in each piece. More white and more likely it will be part of the second or of the third picture. In our case, however, we have to work with nucleotide sequences. So the features that we used was the pentameric frequencies of short-read contigs. Here, as you can see, there are all the steps of the approach we use that are here summarized, and that now I will explain one by one. Okay, as first, we retrieve the initial data that we use to train the model, which, as said before, it's a requirement of machine learning, of a machine learning approach. About that, we know the number of the sequences and the type of sequences of each isolate. And in total, as we can see here, we obtain 146 complete genomes from the NCBI assembly database. So that we had 146 chromosome sequences and 214 plasmid sequences. And as said again before, we will take advantage of their short reads. Of this data, we will perform some analysis to assess the quality, to determine their quality. So as first, we check the diversity of the isolates. We use pop-punk and MLST. In the picture, we have the trees, which have been built using the core genomes, while these other are clusterization based according to the accessory genomes. Pop-punk, sorry, it moved, okay. Pop-punk identified, as we can see here, 28 clusters, while MLST identifies 33 sequence types. The clusterizations were similar, so as first we assessed that the methods were consistent. And then also among this variety, we can also observe that several isolates, like here, were part of a specific group. In this case, pop-punk identified them in the cluster number 1, while in this other case, they were identified as a sequence type 2. They also seem to be redundant. However, since we had big varieties in the end, in the complex, we decided to keep all of them. But however, we also investigated into the plasmid diversity. So we tried to determine the plasmid diversity using mashtree. Here we have a clusterization which have been obtained with using plasmid, the plasmid content of 111 isolates, which are those which contain at least one plasmid sequence, and they are colored according to the sequence type. The plasmid content, also as we can see here, is quite diverse. We still have, however, also some isolate of sequence type 2, which are colored in yellow, which are similar, but some of them are also different one respect to the other. So we could in the end assess that we have also enough varieties among the plasmid sequences. After this, we also asked if we could actually differentiate between plasmid and chromosomes using the chimers, specifically because with the pentamers, because our initial assumption was that we can actually discriminate between plasmid and chromosome sequences using looking at their pentamer compositions. So in this dendrogram was built using a mashtree with a chimer size of 5 for the pentamers. And if we focus here at the bottom, we have this bottom bar where we can observe that the plasmid sequences actually cluster together and differently from the chromosome ones, which actually plasmid cluster together too, as we can see here, the blue part. So again, consequently, we proved that we can split actually the isolates according to their pentamers. And so we can prove our initial assumption. So at this point, now that we have the data, we move to the next steps. So again, here they are summarized. And what we did was, starting with our genomic sequences, we simulated the reads using WGSim. Once we have the reads, we perform a de novo assembly with space so that we could get the contigs. Then we map the contigs against the reference sequences using BWA, so that in this way we could have the label about the origin of these contigs, if they came from a plasmid sequence or from a chromosome sequence. And after that, we compute the pentameric frequencies of the contigs. Now, at this point in machine learning, we need to generate a training and a test set. So to train our model, we had to split the set of data, which consisted of around 12,000 chromosome contigs, as we can see here, and around 800 plasmid contigs. From this pool, we extracted these the contigs of 5 isolates, which consisted in represented our independent test set. And then we split the remaining part in a test set, which consists in the 20%, and in a training set, which is the 80%. Here we can have an overview of the algorithms we use, which are super vector machine, random forest, naive Bayes, logistic regression, and decision tree. Here the evaluation matrix we use to compare the models. Here the categories with the plasmid, which has been defined as the positive one, while chromosome as the negative one. And then the threshold for the label assignment that was set up at 0.5 in both categories. So this is the first result. And here we can observe that all the algorithms actually predicted the plasmid class poorly. So if we look at the graph on the left side, which are actually is a graph that collects together the rock curves, we can observe that actually an ideal curve should identify an area equal to 1. It should have this kind of shape that I'm just drawing with the mouse. Consequently, a curve that identifies the biggest area corresponds to the best model. So in this case, we can already have a ranking of the algorithms and identify supervector machine and this one and random forest as the best model. While if we move actually the attention to the other graph, we have several parameters which are displayed for each algorithm. And we can observe how actually the chromosomes are really well predicted if we look here at the specificity values, while on the other side, the plasmids are almost completely not identified if we look here at the sensitivity value. So to look at the probability distribution of the predictions more in detail, we try to improve the performances. We generated this graph and we observed that the majority of chromosomes, as we can see here for random forest and here for supervector machine, actually the chromosomes are correctly identified. And they are also predicted with a really high probability value, almost 1. So, but as we can see here, it's not the same case for plasmids, which are really badly predicted. So in our case, we try to take advantage of this and we try to improve the prediction, changing, for example, the threshold for the label assignment, as we can see here. So chromosome predicted with a probability of 70% would be classified as chromosomes. By those which were predicted with a prediction value lower than 0.70 were not predicted as chromosome, but were predicted as plasmid, so that they could be classified actually in the right category. Another approach that we use to increase the predictions at this point consists of using a balanced training set. Before, we use a training set of data which represent the actual distribution of the two classes. However, the two classes are represented in an unbalanced way, as we can see here. So, since a balanced training set should be favored in machine learning, we try to improve the prediction generating a balanced training set, as we can see here. So, this approach of reducing the number of chromosome observation were performed with different approaches, which are summarized here. But the main important part is that in the end, we generated this balanced training set. And at this point, we selected again the algorithm into which fit the balanced training set. We excluded actually decision tree and the logistic regression. This exclusion was done because actually they performed really bad before. And also we exclude the naive Bayes because since it takes into account for each observation the probability of being part of one class or of the other in a real distribution. And since chromosomes are more abundant in a real distribution, it was actually meaningless applying a balanced training set where we reduce the chromosome observations. So we decided to focus on random forest, as we can see here, and SVM. On the x-axis of this graph, here we can see an unbalanced training set and the 3 balanced training sets. If we focus on the F1 value, we see a clear actually increment, as we can see here in random forest or here in SVM. We see a really increment with a balanced training set compared to the unbalanced one. And this actually confirms consequently the expected improvement of the approach we follow. After this also, again, in an attempt of still trying to increase the performances, we increase also the number of observations. So in our case, the bottleneck was represented by the number of plasmid observations. So we needed to retrieve other plasmid sequences. To do this, we used the PLCB repository, from which we extracted, we downloaded 129 plasmid sequences that again we put together with all the other previous observations. And again, we recomputed the training and the test sets, both unbalanced and also the balanced ones, and we evaluated the algorithms on them. We can see here on the top that we have here on these 2 top graphs, we have the models generated with random forest, without and with the additional plasmid observations. While in these other 2 graphs, we have the performances of SVM. As we can see, actually, if we compare with the graph, the models, without addition of plasmid and with plasmids, we don't actually observe a really increment in the performances. The only increment that we observe, which is quite relevant, is only this one, which is referred to a supervector machine. So what we decided to do was to consider this model as our selected best model. And in this slide, as final results, we have the comparison about the improvement that we got from the first model that we generated using SVM and the final model that we consider our best models. As we can see here, actually, we have a model. This one was generated with an unbalanced training set. This one instead, it has been generated with a balanced training set with also addition of plasmid sequences. And also, as we can see here, just by the name, we add also slight variation in the threshold for the level assignment to 0.60. So in this way, the model achieves absolutely better performances compared to the first one, to the initial one. So, and this can be, it's clear looking at the F1 value and also looking at the plasmid and chromosome predictions, so that the sensibility and specificity values, which are now more balanced compared to the previous model. In the end, as final result, we tested the model on the 5 independent isolates that we extracted at the beginning, and we compared the performances with other 2 commonly used tools, which are PlasFlow and CBAR, which are used to retrieving the plasmid contigs. And again, here we can see in the bars the sensibility and specificity values, and we can assess that our model actually perform the best way compared to the others. So at this point, we are at the conclusion where we can observe that actually a machine learning based approach is a reliable method for the identification of plasmid and chromosome-derived contigs, because we could generate actually a model with quite good performances. Then the generated classifier can be applied to unseen data within seconds because we don't need databases. So we just need the pentameric frequencies. So it's something really quick. And also, it's this approach is reproducible because we can repeat the same path, the same approach we used before for the generation of other models, species-specific models, because this model is just referred to as Acinetobacter baumanii. But the same approach, as far as we have enough data, could be applied for the generation of other models. And according to this, we also observe that a balanced is favored. However, there are also some limitations in this model. The first is about the chimer size, because other chimer size, bigger, might have higher specificity and sensitivity to discriminate between plasmid and chromosome. It's always a matter of trade-off between the number of data points and features, because if we have a high number of features, really high compared to the number of data points of observation, we can have the problem of the curse of dimensionality. But on the other side, if we also increase the number of features, we could have like some problems in terms of computational demand. So this is actually a problem. The other is that, as stressed before, we can't retrieve individual plasmid sequences, but just if a contig comes from a plasmid collection or from a chromosome sequence. And to solve this problem, actually, we could use a G+, the tool that Sergio Redondo Alonso developed. And the other limitations is that it has a reduced reliability on identification of contigs with antibiotic-resistant genes. In fact, contigs with antibiotic-resistant genes, both if they are plasmid or chromosome-derived, actually it's quite problematic to retrieve them. The predictions are not so specific. So in the end, I want to thank you for the attention. And also I want to leave here my contact details, since I'm looking, as introduced at the beginning, for a PhD position. I'm at the end of my master, and I would like to proceed with my studies in the same field of antibiotic resistances, mobile genetic elements, and also maybe bacteriophages. That's all. Thank you for the attention.